We are going to study about the receptors, classification of receptors and their action. First, receptor is a macromolecule, and most of the receptors are proteins. They may be either inside the cell or binding with the drug, or it will be on the cell membrane. Unoccupied receptors will not be producing any influence. This drug plus receptor forms a drug receptor complex which will produce a response. Signal transduction is the transmission of molecular signals from a cell's exterior to its interior. That is known as signal transduction. Signals will be received by the cells and transmitted effectively into the cell to ensure an appropriate drug response. Drugs here are signals and receptors are signal detectors, and again second messengers or effector molecules are also present in this cascade of events. And receptor, there are functions of receptor. One is recognition of specific ligand molecule. So the receptor recognizes which drug is going to bind with this receptor. Second one is transduction of signal into a response. So it produces a response on the effector domain. Then drug receptor interaction. One is selectivity. Example, adrenaline is selective for alpha and beta receptor. Second one is affinity. Drug should have an affinity with the receptor. Third one is intrinsic activity or efficacy. That means it produces a pharmacological response after binding with the receptor. The drug produces a pharmacological response. The drugs are classified into two classes, that is agonist and antagonist. Agonist means induces an active conformation of receptor proteins. That means the agonist conforms with the receptor protein, but at the same time antagonist doesn't conform with the receptor and it is not producing any response. Now there are four families of receptors: ligand-gated ion channels, G-protein coupled receptor, enzyme-linked receptors, the intracellular or receptor regulating gene expression. So the first one is ligand-gated ion channels. It produces a resting membrane potential and shaping action potential and other electrical signals by gating the flow of ions. So it allows a channel to open through which the ions will be flowing into the cell membrane or outside the cell membrane. Functions: neurotransmission and muscle contraction. This is the diagram of one action potential, and this is the diagram of ligand-gated ion channel. Example here is acetylcholine receptor. There are other examples also like GABA. Here the agonist is binding, and once the agonist comes and binds, the channel will be opened for sodium ions. Sodium ions move into the cytoplasm from synaptic cleft, and here postsynaptic membrane also is shown, and the volume of sodium ions inside the cytoplasm will be increased. And this is the diagram of the structure of one ACH receptor. And here when ACH comes and binds with this receptor, the channel will be open. Sodium ions will be moving outside. Sodium ions will be moving inside, and potassium ions will be moving outside. And this ligand-gated ion channels are having 5 subunits, 2 alpha units, 1 gamma, 1 beta, 1 gamma and 1 delta. And each subunit is having 4 transmembrane domains, that means binding sites. And this is the G-protein coupled receptor. G-proteins are also known as guanine nucleotide binding proteins. It produces effects by the help of 2 enzymes. One is adenyl cyclase and the other one is phospholipase C. There are many kinds of G proteins, GS, GI, GQ, etc. depending upon the ligand which is binding there. The alpha subunit binds guanosine triphosphate and beta and gamma subunits anchor the G proteins. That means it is having three subunits. One is alpha, one is beta and gamma. Alpha is binding with guanosine triphosphate, beta and gamma. They are binding with G protein in the cell membrane. Now here hormone is the drug which binds with the G protein receptor, coupled receptor, and it produces its action first by replacing GDP by GTP and with the help of the enzyme adenyl cyclase, which helps the conversion of ATP to cyclic AMP. Here RS is the receptor which produces stimulation and RI is the receptor drug complex which produces inhibition. Here also the same enzyme is the adenyl cyclase which helps in the conversion of ATP to CAMP. Then here is the diagram for the G protein coupled receptor which is a coiled receptor. Here example is alpha 1 receptor which is a receptor for adrenergic drugs like adrenaline, and phospholipase functions as the enzyme. This is the alpha 1 adrenergic receptor signaling cascade. Then there are varieties of G protein. One is GS, is a receptor for beta adrenergic, GI for alpha 2 adrenergic and ACH, GQ for ACH, and GO for neurotransmitters in brain. And the signaling pathway for GS, that is stimulation of adenyl cyclase